Recent improvements in multi-electrode recording technologies have led many people to work on data analytic methods for identifying neurophysiological interactions among distinct parts of the brain. Our research is motivated by what seems to be a somewhat unusual perspective derived from my lengthy experience as a statistician. My primary research support for more than 20 years has been from the National Institute of Mental Health, and I've most recently pitched this by suggesting new statistical methods could lead to improved treatment strategies involving interventions that are precise in space and time. It seems to me that 50 years from now, treatments for mental health disorders will have advanced far beyond where they are today. But to get there, neuroscience will have to go beyond simple static circuit diagrams that depict interactions among brain areas without describing the flow of information while the organism accomplishes some behavior. This kind of static diagram displays the different brain areas that may be interacting when an organism carries out a task. In the case of this diagram, some rewarded behavior. But the behavior is constantly evolving and brain activity is constantly changing. The diagram doesn't show us at what point in time particular pathways are involved. It gives us no notion of sequencing across time. That is, to make dramatic improvements, the field must not only say what circuit should be disrupted by an intervention, but also more precisely where and when, relative to behavior, disruption should take place. Improved characterizations would use timing relationships, consider both local and cross-area computation, and describe cross-population dynamics. The first of three projects I want to summarize for you very briefly is concerned with spike trains, hundreds of them recorded simultaneously across 6 arrays in about 10 brain areas involved in visual processing in response to visual stimuli, specifically drifting gratings of varying orientations and frequencies. In this initial article, we focused on 3 brain areas, V1, LM, and AL. V1, primary visual cortex, feeds into the higher visual areas, LM and AL. Here are smoothed histograms displaying neural activity aggregated across trials and across the dozens of neurons recorded from each area. Each curve displays a population firing rate function as it evolves over time. The most prominent features of these curves are 2 peaks in firing rate labeled 1 and 2, with the first occurring much earlier than the second. One would think that these evoked responses in neural populations could tell us a lot about the interactions among areas. In the trial averaged firing rate curves, peak 1 seems to occur earlier for area V1 than for the other 2 areas, as we would anticipate. And we would also expect that this evoked response would occur earlier in V1 than in the other 2 areas consistently on a trial-by-trial basis. The same might also be true for peak 2, but the situation then is more complicated. The second peak occurs sufficiently late following the stimulus that it likely receives important input from higher cortical areas. So, can we see a relationship between V1 and LM peak 2 timing on a trial-by-trial basis? To answer this question, we will find the time at which each population reaches its maximal firing rate and compare across areas. To do this, you might think you could simply fit curves to the peak on each trial and thereby compute the peak times, but statistically, that doesn't work very well. The panel on the left, labeled B, shows the resulting times at which peak 2 reaches its maximum in V1 and LM, with each black dot representing the pair of times on a given trial. No relationship at all is visible. On the other hand, a Bayesian approach based on a hierarchical model reveals, as shown in the panel on the right, labeled C, a very strong relationship, with the posterior distribution of the correlation of peak times portrayed in the inset as a representation of statistical knowledge and uncertainty. Also, the dots in panel C are pretty heavily clustered below the diagonal, and according to the posterior distribution, the peak in LM occurs later than the peak in V1 by about 10 milliseconds. A descriptive name for this kind of analysis is evoked spike response coupling, and it emphasizes its similarity to analysis of evoked response potentials, ERPs. In fact, the methodology assesses coupling based on curves derived from selected neural populations that are specific to experimental condition. Notice the much higher firing rates. The resulting population firing rate curves are sharper, especially for the second peak, which typically attains a maximum firing rate similar to that during the first peak. Also, the most common method of summarizing population responses, Gaussian process factor analysis, GPFA, does find peaks on one of its factors, but it doesn't find coupling. It produces a figure much like panel B. The hierarchical model, which we call an interacting population firing rate model, and the associated computational method, was developed by Yu Chen, a student who recently defended his thesis in our machine learning and neural computation PhD program. It provides denoising and covariation of peak timing, as I've shown you, and it includes clustering of neurons and uses time warping for trial-to-trial variation. Schematically, according to the model, spike trains are generated by point processes with intensity functions that depend on one of three templates, that is, their template functions according to neuron membership indicators. Only one template function, the interacting population template, is involved in cross-area communication. The other two are there to absorb the majority of neurons that do not participate. The non-parametric template fitting, along with the clustering of neurons, is carried out for each brain area and each experimental condition. A small number of features, including the times of the two peaks, capture trial-to-trial variation of the kind of covariation I just showed you. Importantly, the method is multivariate, so we can look at graphical structure based on partial correlation. For example, we know that V1 feeds into LM and AL, but these areas also get input directly from the thalamus and from other visual areas, which could be especially relevant to the timing of peak 2. As I said, by the time of peak 2, feedback loops are likely to be operating. So what happens when we examine the correlation of timing in two areas, such as LM and AL, after conditioning on timing in the third, in that case V1? Before conditioning on timing in the third area, panel A shows very high correlation of the peak timing of LM and AL. Panel B shows a little smaller, but still very high correlation of the peak 2 timing in V1 and AL. Panel C shows that after conditioning on timing in V1, the correlation of timing in LM and AL is somewhat reduced compared to what's in panel A, but it's still very large. In contrast, panel D shows that after conditioning on timing in LM, the correlation of timing in V1 and AL is dramatically reduced compared to panel B. There's no longer any visible relationship between them. These are interesting relationships, which we have replicated in a different mouse. And of course, plots are all easily obtained from the posterior distribution of the covariance matrix for the trial varying features. Let me now turn to the second project, torus graphs for multivariate phase coupling, where we analyze local field potentials that are oscillating. As you know, oscillating local field potentials have been observed under a variety of circumstances, and there are plausible mechanistic network models to explain them. One leading theory is that they facilitate communication across brain areas by forcing populations of neurons to spike at nearly the same time. Because neuron membrane voltage decays fairly rapidly, such nearly synchronous spiking could increase the firing rate of a downstream population. Earl Miller's lab at MIT has been especially interested in oscillations during learning and memory tasks, with several papers focusing on the interaction between prefrontal cortex and the hippocampus, which are widely considered to be crucially involved. We analyzed some of Brinkat and Miller's LFP data, bandpass filtering it as they had done at 16 Hz. We then selected a particular single point in time on each trial at which to measure the phase from each electrode, and looked for phase coupling, meaning a fairly consistent phase difference across repeated trials which would indicate coordinated activity across the brain areas. The standard measure of phase coupling is called phase locking value. It's intuitive, and as we show in the supplementary material of our paper, under special circumstances, it does estimate a natural analog of correlation for angles, which in statistics we call circular data because an angle specifies a point on a circle. However, phase locking value is a pairwise measurement. While Brinkat and Miller had 24 electrodes across PFC in 3 regions of the hippocampus, CA3, DJ, dentate gyrus, and subiculum, so we wanted a multivariate method, which would give us a more complete picture of interdependence as provided by a graphical model. That is, we wanted a placement of edges connecting the 24 nodes shown here on the right, where the edges indicate statistical dependence. We can construct a set of nodes and edges using phase locking value, but that doesn't give us a probabilistic graphical model, meaning that the absence of an edge using phase locking value does not correspond to conditional independence given all the other nodes. We wanted instead a general circular analog to partial correlation. In some circumstances, a Gaussian graphical model based on the multivariate normal distribution might be satisfactory, but not here. Although these experiments have a well-defined start time on each trial, that doesn't do much to set the phase, so the phase angles across trials are fairly uniformly distributed. To build a graphical model, we have to respect the topological distinction between a circle and the real line. Here's some data simulated to look similar to real data. Real data are shown in the small colored picture to the right, where 2 phase angles were drawn repeatedly, with each being roughly uniformly distributed but with a pretty consistent phase difference. x1 is leading x2. Note the seemingly weird data points in the upper left corner of the plots. When x1 goes a small amount, c, past pi, 180 degrees, pi plus c is replaced by minus pi plus c. Meanwhile, x2, lagging behind x1, hasn't yet gotten to pi. So it's easy to explain what's going on up in the left-hand corner. But the weirdness is an indication that standard xy coordinates are not really appropriate. It's more natural mathematically to consider the product of 2 circles, which is a torus, with one angle identifying rotation in the direction shown here of the blue arrow, and the other angle identifying rotation in the red direction. After smoothing the simulated data to get the color density representations shown here, we see that the weirdness in the plane is gone when it appears on the torus. One of the students made this animation to show more clearly what's going on. The red dot indicates corresponding points in the 2 representations. The torus is clearly a more elegant representation, and to get a 24-dimensional analog for phase coupling analysis, it is statistically natural to develop an exponential family on the multidimensional torus, which we call a torus graph. More specifically, we consider the full exponential family on the multidimensional torus having all 2-way interactions. A torus graph is a subfamily having some specified set of 2-way interactions, which can be represented as edges between the nodes of a graph. The 2 PhD students, Natalie Klein and Josue Oriana, did a very thorough job of establishing the properties and methodology, so there are many interesting aspects of the work. But the key point here is that the absence of an edge in a torus graphs corresponds to conditional independence given all the other nodes. In other words, torus graphs are probabilistic graphical models. Again, a graph based on PLV, the pairwise measure, does not have this property. The portion of the linear probe used by Brinkat and Miller that recorded from area CA3 had 5 contacts. The torus graph we obtained recapitulates the linear arrangement, with each electrode exhibiting Markovian conditional independence from non-neighboring electrodes conditionally on its neighbors. The PLV graph makes it look like every electrode is dependent on every other electrode. When we consider all 24 electrodes together, again, the torus graph gives us something that's consistent with known anatomy. Prefrontal cortex depends on the dentate gyrus only through its dependence on CA3 and the subiculum. Finally, the last project again considered cross-population coupling, but this time based on local populations that generate the local field potentials, that is, the current sources. According to basic electrostatics, the current source density, or CSD, is the negative Laplacian of the LFP, and the first methods of estimating CSD simply applied numerical second derivatives to the LFP. More recently, it was recognized that CSD estimation is an inverse problem that can be solved using the forward model that maps the CSD to the LFP. This version of the standard forward model comes from our paper where we used A to denote the forward model linear operator. Our statistical perspective led us to model the LFP as the sum of the forward model applied to the CSD plus noise. Based on our experience with LFPs, we then took the CSD to be the sum of three components, a non-stationary transient together with a two-component Gaussian process. Standard forms of Gaussian processes describe fluctuations using a single time constant parameter. Here, we forced the two components, the two Gaussian process components, to have very different time constants, making one of them relatively slowly varying and the other more rapidly varying. The first was designed to capture trends, and the second, the oscillatory fluctuations we were interested in. So this is a Gaussian process current source density model, and it can reproduce LFP spectral densities extremely well. The paper provides reasons to believe this approach is more accurate and more general than other existing ways to get CSD. And in auditory cortical data, we found 10 Hertz phase coupling across cortical layers in two separated columns of cortex that were not apparent in the raw LFP. So those are my three examples, and as I've reflected on what binds them together, I've observed that in each of these three cases, interactions across neural populations seem to become apparent when interaction and population are defined appropriately. The interacting population firing rate model defined interaction in terms of peak timing and used clustering to select relatively homogeneous populations of neurons, thereby revealing relationships that could not be seen using naive methods. Torus graphs define multivariate phase coupling as a form of interaction, producing relationships unavailable from the pairwise measure. And in the case of Gaussian process current source density estimation, the relevant populations were those we found to be generating the currents. One of the things that makes LFPs hard to work with is that they reflect a superposition of differing voltage patterns from many distinct current sources. Our GPCSD estimation amounts to deconvolving the LFP into spatially distinct components. Also, when I mentioned we found phase coupling across cortical layers, shown here as superficial, medium, and deep on two different probes labeled lateral and medial for their locations in primary auditory cortex, that, of course, was again using torus graphs to define interaction. Importantly, we were unable to see this kind of pattern when we examined the raw LFPs. In all these cases, considerable experience and wisdom within the field of statistics informed the design of these approaches. The examples also illustrate the three scientific goals I started with: using timing relationships, considering both local and cross-area computation, and describing cross-population dynamics. And when I said we should consider both local and cross-area computation, I had in mind the substantial structure within areas reflected in particular by cortical layering, shown here, that does seem to carry out local computation. The three examples did, in a certain sense, illustrate these three aspects of neural computation. The interacting population firing rate model used timing relationships. Torus graphs describe cross-population dynamics in the form of oscillating circuits. And Gaussian process current source density estimation considered both local and cross-area computation. Local interactions occurred within one of the two probes and are not shown in this figure. But in terms of what I meant the field of neuroscience will have to accomplish, what we did in these papers amounts to taking baby steps along that path. One of the most frustrating issues is the need for good statistical descriptions of cross-population dynamics that can be used in conjunction with mechanistic descriptions. Put differently, we should ask, where are the synergies of statistical and mechanistic mathematical modeling? This was the point of a set of discussions, including 2 international meetings, that led to the review paper cited here. I like the paper a lot, but I found the effort nonetheless disappointing. Not only were there few good examples of the synergy I've been looking for, there were few concrete ideas for bringing together these 2 sub-disciplines of computational neuroscience. One simple way to start, which I hope to pursue with a couple of students, returns to the commonality among examples I just pointed to and poses an obvious question. Under what circumstances, what assumptions, can that observation be made rigorous and be verified theoretically? We could, I presume, specify assumptions purely statistically. In the case of the interacting firing rate model, we could assume that we recorded samples from homogeneous local populations driven by firing rate functions. We defined interaction using peak timing, which led to well-defined directional timing relationships across areas. But while peak times are intuitive, using them is really a statistical convenience. And we might want to think more generally about forms of dependence between time-varying functions that are driving separate neural populations. There's all kinds of dependence we could dream up statistically between random functions, but they ought to be possible in neural systems. So it would be much more interesting if we could connect to that large body of work that gives mechanistic descriptions of neural population dynamics. But then, our statistical models would not simply be describing variation in data, they would also become part of a mechanistic explanation. And within the field of statistics, this sounds like a fundamental shift in the way we think about the nature of statistical modeling. The nature of statistical modeling is something that statisticians have discussed for many years, and I've added my own comments in several places. I would like to draw your attention especially to a recent contribution, which acknowledges the commonalities of statistics and machine learning, including artificial intelligence, but then tries to specify the continuing value of this perspective advanced by the field of statistics. Common wisdom, correctly in my view, places statistical models at the center of modern statistics. However, with all the thought I've put into this across many years, my appreciation for the value of mechanistic thinking in statistical modeling is not something I've articulated. Concerning the possibility that models provide explanatory mechanisms, statisticians remain agnostic and skeptical, whereas neuroscientists are boldly adventurous. I've come to realize we need both attitudes. And what's shaped my thinking, as much as anything, has been my experience over the past 10 years teaching a basic graduate course in computational neuroscience. I call the course Statistical Models of the Brain with the hope of emphasizing that it is not a course in data analysis, but rather a course that surveys many leading roles played by statistical models in explanations of how the brain works. By the way, if you go to that website, you will find that there is a video for nearly every lecture, and in fact, the videos are very short, they're concise, so you may want to have a look. Witnessing the way neuroscientists use statistical models for explanations makes me think there may well be some benefit for statisticians to look more favorably on the notion that models can, in some sense, specify mechanisms, at least in certain circumstances. But what circumstances? Perhaps I'll figure out something coherent to say about this and write about it. We'll see. In any case, I do hope some of you might consider this a topic worth pondering. Thank you, and sorry I couldn't be there.